Hi, my name is Michael Polymenis. I'm a professor in the Department of Biochemistry and Biophysics and a member of the graduate program and also of the graduate program of genetics. So you're seeing this video because now that you're starting your career as a graduate student, as a PhD student, you have to find a lab to do your thesis project. But before you do that, you have to try out a few labs in a process we call rotations. So this video will tell you a few things in general about the general questions and philosophies that go into picking a lab for a rotation, and some more information specifically about my lab. And we're going to finish with a small tour of my lab so you can see how it looks like. And if after all this you want to talk to me in person, you can always reach me by email or visit me at my room 333 in the Biochemistry and Biophysics building where I'm located. All right, so I will start by sharing my screen. I have prepared a few little slides with just a few questions and points that I want to make. So let me share my screen. So what is a rotation? It's more like it really resembles dating. You are trying out a few different labs. You're going to do at least 3 rotations. And before every rotation, you have to submit a ranked list of 3 labs and that you are interested in, and a matching will happen, and you're going to be assigned one of those 3 labs and that you picked. And hopefully, out of the overall, you're going to do this 3 times, and through after the whole process, you will have found you would pick a lab that you think you are a really good fit, you feel comfortable with, you are excited about the research, and you're ready to go. All right, so it's a very important process, in other words. But before you talk to the PIs and pick specific labs in your ranked list, there is some homework that you can do and really narrow down that list. First, of course, you visit each lab's website and read about their research, what they do. I mean, does the problem excite you? But before even you get to the specific scientific question that might be of interest to you, there are some general categories of experimental work. We're all experimentalists in these programs, so you gotta do you're gonna do things with your hands, but there are different things that you can do with your hands, depending on the lab you choose. Are you more into instrumentation? You like to take big sophisticated instruments apart or build new ones and put them back together. Maybe you like to look at living organisms, from cells to animals, plants, and how personally I like to see how cells grow and divide, for example. Is that what turns you on? I mean, you have to find what your passion is. Maybe you're more passionate about looking at purified molecules in a test tube, how they look like, how they interact with each other, what kind of reactions they do, chemical reactions. Or maybe you really enjoy writing code in front of a computer, so to help help others solve complex biological problems. These things are not, of course, mutually exclusive. You can like a few of these things, but depending on what you like, I mean, you can really, if you really like living organisms the most, it's natural not to join an exclusively computational lab, because you won't have much of an opportunity to look at living organisms, for example. The other thing you can see and find out on your own before you even talk to professors about the possibility of doing a rotation in their lab is to look at what the lab publishes, how often, where, what kinds of papers, and also find out if the lab is funded. So you can find all that on your own. So first, let's start with publications. Why do we care about publications? Because this is the end product of what we do. You have to generate new knowledge, and you have to publish it. If you don't publish it, pretty much you haven't done it. So, and there is the old maxim, publish or perish, which governs all of academia pretty much, including you as a grad student. Okay, you do have to produce published work to be awarded a PhD. So how do you find these publications? Well, it's very easy. So you can look at this website. So let's pick one. Let's say Google Scholar, for example. So let's click on it, and you type, let's see, type myself, okay, Polymenis, Michael. There are not too many Michaels. Yeah, we can pick this, and you can see, for example, this is the profile, and you can find specific papers. And let's make this a little bigger. And you can sort by date. It's probably advisable to look at specific dates. Let's say the last 6 years, how long a PhD lasts. So don't go too far back. You have to be up to date. So you do this, and you see the things I have published, in the last 6 years, and you can click on any one of these papers, and you will see if the abstract, from the abstract you can read it, and you can see if that excites you. Another important thing to look at publications is citations. For example, how many times the paper has been cited, so here, and that will give you an idea if this is a better paper or not. Also, from the list of authors of the publication, let's click on this one, for example, you can tell if the lab was a senior contributor to this work. So usually the professor will be at the very end, and he or she will be the corresponding author. But another very important thing is you can see individual people, the authors, and you can try to imagine yourself as a grad student now being one of these authors. And if you're happy with the picture, taking out one of these names and putting yours in, and preferably as high up as possible, that will be, and if you're happy with this outcome, then that's a good thing, that you can imagine yourself in that lab producing something similar and moving on. But the bottom line is you really have to do your homework and see these papers, see what the lab publishes, and picture yourself in that lab, in the output, in the product of that lab. All right, so that's one thing, but there are more things that you can look at. Let's go back to the presentation, and as I said, you have to look at the quantity, quality, and the authors of every paper. So what the lab publishes, and try to imagine yourself in that lab, in the publications they produce. Now, the next thing you can find out is funding. Okay, so why is funding important? Because as I said, the end goal of our enterprise in academia is to produce new knowledge and publish it. But ours being experimental science, you actually need to have money to support your research. And if you don't, then your PhD will be difficult to do, much more difficult. So you look at the funding of its lab, and again, it's very easy to do. So you look at these repositories and that have been put together by federal agencies such as NIH or NSF. So here, let's click again on this one. The first one, for example, from NIH, and you can click on it, and you can put a name. Again, if you put myself, you're gonna find out if I'm funded or not from the NIH. But there is more nuance to it, so you can find out how long. So, for example, this is my active. You can click on active grants. It's going to tell you this. So let's say we can look at this grant, and it tells you again the abstract of the grant. Okay, so it describes the work we're doing. These are all publicly available, and you can do this on your own for every person you can. So do your homework, and you can do this for NSF, for NIH, for USDA, for all other agencies. After you do your homework and you've seen if you're interested in the work based on the website that you looked at, you decide that the lab is publishing well and it is funded, then you should talk to the PI and talk there in more detail. Some of the things you may want to talk to the PI about is a possible thesis project. So these are the long term, for your PhD. but also specific rotation projects. So we discussed about funding, and then you should talk to the PI about the expectations. What are the expectations of the PI when you join the lab? For example, I can tell you that my expectations are that you definitely need to you have to be passionate about your project. You have to put the hours to do the work that's necessary, and you have to read about your project. I mean, read the current literature. You have to go to seminars and basically soak it all in. The way I see a PhD is a fantastic opportunity. Somebody's actually paying you to play in the sun and just enjoy the opportunity to do science and learn something new. This is an amazing opportunity. So don't see this, the PhD in my mind is not a collection of LinkedIn skills that you can put in your profile and hoping to get a better job. Of course, with a PhD, hopefully you will get a better job, but don't lose the passion that should come along, okay? And the motivation should be there. So it should all come from within. I cannot motivate anyone. You have to be motivated on your own to do the things you need to do in the lab. And I will tell you more about it as we give you the little tour in the lab later. Talk about the PI, talk with the PI about the mentoring style. You know, my lab is small, it's not big, and that could be different, you know, how the PI interacts with people depending on the size of the lab. I try not to be a micromanager, so you're going to be on your own, and you know, not on your own, but you're not going to be. I'm not going to be looking over your shoulder, you know, every hour on the hour to see how you're doing. But I do expect things to get done, and I do expect you to talk to me if you have a problem and try to solve it anytime. My email is open 24/7, so. I like to do experiments in the lab, and so you will see me a lot in the lab, like everybody else, and also in my office. Another thing you need to talk to the PI about is the average time to degree. I mean, if the average time in the lab is 8-10 years, I mean, is that something you're willing to do? Okay, I can tell you the average time in my lab is no less than 5 years and no more than 6 years, and I'll give you the information on that. Another thing you need to ask the PI about is what happens to the former students in that lab. Did they advance in their careers? Where are they? So these are simple questions, but you have to talk with its PI that you are interested in rotating through. I mean, you have to talk to them about that. Then you do your rotation, and you can change your list. You know, you pick 3 labs for rotation 1, then you can pick 3 different labs for rotation 2, and so on and so forth. And so have an open mind. And after you do all your rotations, you will see what kind of mentoring style is best for you. I mean, do you really want a constant, you know, supervision, or you are more comfortable with a little less of that, be a little on your own, and then get the feedback in a more regulated way? Again, do you like big labs or small labs, and what is the lab environment? You know, having a party atmosphere may not be the most appropriate lab environment for your thesis work. On the other hand, if it's too oppressive, I mean, also that's not very good. And I stole this from this interesting article that a student published, so you may want to look at it. All right, so now we can go to my lab and see something more specific. So again, visit, let's say after all this, you're still interested in my lab. Again, go to my lab webpage and look it up. You're gonna click on the webpage and you're gonna see something like this. All right, let me move myself a little bit. So there's going to be a little overview of what we do, and we use budding yeast as a model system to study how cells grow and divide. And why do we use yeast? You can read the reasons here, but basically yeast is unique. It's a eukaryote, so whatever we learn in yeast, hopefully we can translate it into humans. And it's a microbe, so it can grow fast and cheap and easy. It has excellent genetics. We can make any change we want. It's probably one of the easiest organisms to work with. But the reason is but the main reason for us is it's unique for the problem we study. We study, again, progression during cell division. How do cells know when to initiate a new round of cell division? How do they know they have grown enough? And budding yeast makes a little buds shown here on this picture. When it makes that decision, it's really unique. No other cell will do it. I mean, you look at our human cells, and they look the same, whether they have initiated a new round of cell division, making new DNA or not. But budding yeast lets you know by making this little bud, which eventually will grow to become a new daughter cell. So you can just look at them and know, and you can score the key decision. And for us, this is very important. So for all these reasons, we use budding yeast. If you want to know more about our research, you can again look at our papers. Some of them are listed here. All of them are listed, on PubMed or other sources. And how about people? I mean, some of the questions I talked about earlier. Well, so as I said, my lab is small. This is where we are right now. So we have a group of 4 or 5 people, and you can find out who my previous students were, how long they stayed in the lab. As I said, the typical PhD duration in my lab is 5 to 6 years, and what they have done. Some end up in academia. Some are still, doing in their postdocs. For example, my last student is now at the Fred Hutchinson Cancer Research Center doing a postdoc. And you can find all this information. All right, so you now at this point, let's say you are interested in doing a rotation in my lab, contact me. This is my email. The address again is Biochemistry and Biophysics building. I'm on the 3rd floor in room 333. So good luck in your journey that you just started, finding a lab for your PhD. And if you're interested in what we do, you want to talk more about it and possibilities of rotation projects and our thesis projects, come and find me. All right, good luck. I will stop here and I will continue again in a minute in a virtual tour of the lab. All right, so let's take a little tour of the lab. This is where people sit usually. So we have windows, and you do basic experiments. We are really a molecular biology lab, so we're going to see very basic, simple things. We grow our yeast on these plates, and with little pipettes, which look like this, for example. These guys, you move very small volumes of fluids from one little tube to the next. So nothing super fancy. This is my bench, okay. Then we have some, as I said, really basic, simple equipment for molecular biology, storage facilities. This is kind of interesting because we have several simple microscopes, nothing too complex. This one has a micro manipulator, which you see attached here. And with that microscope, we generate new strains, new yeast strains. Yeast, another power of yeast as a genetic model system is that you can isolate the individual products of every meiosis. And that's quite useful, and we use it when we use this microscope that I just showed you. Whenever we cross 2 parental strains, we generate the progeny. It's a diploid cell, and then we use it to undergo meiosis and to get the gametes and get the combinations that we really care about, and that's how mostly we construct our strains. Yeast, although this piece of equipment that you see here is not in use right now, but it's a fermenter, and yeast is a microbe, so it can grow under very precise growth conditions. At our will, really, we can dictate how fast it will divide when it's grown under these conditions. And so if let's say you join the lab for a rotation, you will be expected to do basic molecular biology experiments, generating maybe a construct, expressing the protein, constructing some strains, and you validate, you monitor your protein, or you monitor your DNA with very simple gel electrophoresis, approaches, very simple basic equipment. So this is our gel bends. But the most specialized technique we use, and we're probably one of the best labs in the world in doing this, is to isolate highly synchronous cells for our cell cycle analysis. So we use this special centrifuge where the cells are actually in this yellow chamber here. It's a continuous flow centrifuge, so media is pumped in and comes out. The cells don't pellet, they just spin in this centrifuge, and they stay in suspension in this yellow chamber. And we adjust carefully how fast we spin them, or how fast we pump media in the centrifuge, and we can wash out the babies, the newly born daughter cells, and they are super highly synchronized, 100%. So synchrony, all of them early in the cell cycle, and then we have introduced all our changes, and we can see how they divide. We take them out, and we put them through these machines that measure precisely their size, how big they are, and we can monitor their growth as a function of time. We take samples out, and we run our gels, and then we see the molecular changes we expect. All right, so this is a typical molecular biology lab, and I have to say, you really have to be part of it. Or any lab you join, you have to be part of that lab. It smells nice to me. I like to touch things, how they feel. I like to see things, how they look like. If I have transformed some cells, I want to see some colonies. The first thing usually I'm going to do when I come into the lab is to pick up my plate and look at it and see if it has grown or something. So it's this idea that you have to have the feel for your organism. That phrase was coined by a very famous female scientist, Barbara McClintock, a Nobel laureate. And that gets to happen the more time you spend in the lab. So how do you know that you're a good fit for a lab? You have to feel that passion, that excitement that I mentioned. And how do you know you have it? Well, let's say if your heart rate goes up before you develop a gel, and you want to see your protein or your DNA, if your heart goes up, then probably you got it. I mean, you are excited, but you want to see what happens. You don't see it as something that you just you punch a list and you have to get through it. The other thing you can, at least from my personal experience, I had done some industrial internships as a student, and I was looking always at my watch, is it time to go home? Well, when I'm in this lab, I never look at my watch, is it time to go home? I mean, this is where I want to be. So through your rotations, you're going to experience such things, hopefully, with some lab, and then if you do, you're excited about the outcome of your results and all that, and the questions you are asking, then that's the best lab for you. So with that, I will close, and again, good luck on your journey.